Thank you, Martha. So a few weeks ago, 3 weeks ago actually, I received the initial agenda for this conference, and I asked Martha, is no one talking about light or melanin? And she said, nope, but I have room for you. So here we are. And this will probably be the most provocative talk you'll hear these 2 days. I attempt to lift the veil, if you will, to show you some of the role that light and the melanin biophysics and biochemistry and biology plays in Parkinson's. And so, here we go. These are ideas that I developed with my good friend, Logan Duval, who Martha knows well. We've been working together for over a year, and our ideas and meetings and discussions and podcasts and so forth have really elevated and accelerated how we understand what's going on in cancer and also in Parkinson's. So here it is. I am not a scientist, so I'm going to apologize right now. Some of my statements and connections will absolutely lack data. But I'm trying to be as creative and an understanding of all that we've come across and learned. We're connecting the dots that we have learned. None of this is our own, but we've learned from all the great, brilliant people that we well know. There's several in the room here. And so some of these you will recognize, some of them you may not. Roberto Becker is on the right of Martha, and Jack Cruz, and May Ho is on the bottom left. She's an amazing scientist as well. And of course you have St. Georgie and Barry and Roland van Wijk, another Dutch incredible researcher, and that's Logan on the bottom right. Fritz Pop is on the top here, and then there's Alexander Gurwitch, who did incredible experiments a long time ago with onion roots experiment. This is Gilbert Ling, and then there's also, I want to pay attention to Jim Humble, Gabor Shumiaye. Martin Paul has done amazing work in EMFs, and Dr. Boros in deuterium, Dr. Jacob in DMSO, which is a very important substance as well. Nina Teicholz in fat, and Michael Crawford on the right, another British person. I don't know if you know him. He did incredible research on DHA. And then, of course, we have Hans in the room. Stephanie Seneff spoke last night. Morley Robbins has taught us all a lot about iron and copper metabolism. Peter Duisberg, I thought, was also very interesting for me. Fred Kumaro is somebody that has done incredible work. He has passed, I think he's over 100 years old, but he did really beautiful research on cholesterol. Roman Zuberov is a scientist in the Karolinski Institute who has done a lot of great work on deuterium research. And then there's Uwe Raskanov, another cholesterol researcher. John Ioannidis, he is an amazing researcher from Stanford that really has, and I'm sure you're very well aware of his work.
Everyone should be aware of what
he should. We should. Yeah, he's incredible. And then there's Nick Lane, another Brit who has done amazing work. He in 2008 wrote a book, and in that he shared that the mitochondrial inner membrane has a equivalent of 30 million volts on it. So it's astonishing if we understand the electrical aspects of how our bodies function. So let's go there, the human body. So if we are electrical, and we emit light, and we also emit magnetic fields. That's the biofield we all hear about. We're largely made of water with some carbon, a few minerals, and we are regulated and fueled by sunlight and maintained by dissolved gas in the form of nanobubbles. If we acknowledge this reality of our structure and function, then we can guide therapeutic up.
Yeah, it's just hard to hear.
I don't have a loud voice. I don't know. Can we do the microphone louder? I don't know.
It's not a mic, it's not a, it's only a mic.
It's the heat. Yeah, okay. I'll try. Thank you, Meredith. Okay, so the energetics of Parkinson's, what do I mean? So we, Logan and I, define energetics in this context as the photobioelectromagnetic array of the quantum dynamic physical forces and frequencies that determine the state of our bodies. It includes fundamental principles such as how we process and transform energy and the universal nano infrastructure, glycocalyx, and its associated endothelial surface layer, which lines all cells, cell membranes, and most tissues. Maybe all, I don't know. So the photobioelectric effect we define as the absorption and emission of light and the generation and conductance of bioelectricity in a living organism or biological system triggered by sunlight exposure and metabolic water synthesis. It conceptually connects light-induced processes with bioelectrical phenomena in living organisms. The harmonization of all this is what represents health. So the powers of melanin. So I have some notes because there's important pieces and I don't want to miss any part of it. Neuromelanin. Oh, I have to say this first. So the pigment power. Silicon Valley's hottest substance right now. Researchers suggest that the superconductive and semiconductive properties of melanin can be harnessed to improve sustainability, innovate bioelectronics, and advanced computer technology. So this is the main substance that they're trying to create and use in the future. So it's okay. The neuromelanin is a dark brown intracellular pigment that has long been associated with Parkinson's. It's found throughout the brainstem, but most concentrated in the substantia nigra and the locus coeruleus. It's a group of nuclei that's part of the reticular activating system, which you, I'm sure, all know. Melanin is in our body, it's essential because it can conduct charge, and it can both inhibit and promote electric flow. That classifies it as a semiconductor. The molecule can switch between resistive and conductive states, depending on the amount of water in the environment or the hydration state. Now, why is that important? If melanin can downregulate, and this is data all the way from Roberto Becker's work, which some of you have just read, this was published in 1970. If you can downregulate a DC charge to 1/1,000,000th of an ampere, you can provide the exact current that is needed to regenerate tissues. The downregulation can only happen if melanin is hydrated with deuterium-depleted water, that metabolic water made by our mitochondria. So the DDW, but that's what we call deuterium-depleted water, that insulates the melanin, and then it provides the right environment to insulate and, or if it's dehydrated, to alter function of the melanin. So what makes melanin so unique is that it's able to convert light into usable energy, just like chlorophyll in plants. Melanin has been shown to absorb a broad spectrum of light, particularly UV, visible, and infrared radiation, also gamma radiation. This means that it's actively responding, absorbing, concentrating, and interacting with the environment. It can also separate charge. And when it's hit by sunlight, it will split water into hydrogen gas and oxygen gas and electrons. So by managing oxidative stress, melanin can also protect us from free radical damage. Additionally, it's a heavy metal chelator and detoxing agent. So that makes you think, okay, if there's a lot of mercury in the system, melanin takes that up. Is that contributing to the dysfunction of melanin down the line? It's all possible. These are all areas to be explored, I think. So it optimizes really truly how our bodies respond and interact with light. Now, melanin is found in all of our sensory organs, and we find it in high concentrations, specifically and it forms a sheet, if you will, in the retina, in your inner ear, and of course in the brain, we know that part, but it's also in other tissues. It's in the vagus nerve, it's in the spinal cord, it's in the heart, ovaries, adrenals, and arteries also contain melanin. The dehydration, so what causes the destruction of melanin? Blue light and non-native EMF are at the heart of the issue. And blue light, I mean isolated LED, artificial light at night types of exposures. And once that you dehydrate melanin, it no longer processes the energy or conducts the electrons in the way it should and that we want. And so everything changes. And as you well know, there are these processes that either neuromelanin can be protective and or it can be a big problem. And once this process starts, then oxygen no longer becomes as useful to us. We can't make enough metabolic water, and lots of downstream effects, including sleep apnea and so forth. As I just mentioned, when melanin breaks down, dysfunction happens on all fronts. This really leads to instability, and a lack of UV light and infrared light is really very much part of this. It's the other side of how we're supposed to manage this. And it really seems to correspond with a decrease in dopamine and in all of the synthetic pathways that we have. There's data, and there's much more, but I didn't have the time to pull it all out. But melanocytes actually sense blue light, and they get stimulated by blue light, which is why women will often with darker skin get melasma, or melanoma is the other well-known effect of that. So there is a lot of data in Parkinson's as well that the neuromelanins really do play an important role, and there is a unclear assessment, I think, what causes neuromelanin to go from protective to actually toxic, and we think it's the light. And then, of course, bright light collapses the glycocalyx. We learned that yesterday. That's another aspect of it. So tyrosine and tryptophan activation is, this is a beautiful graph that was made by Alexander Wunsch. She's a German scientist has discussed many beautiful presentations on light you can find online. But what he highlighted to all of us is that these aromatic amino acids absorb, meaning are activated at UV light spectra. And so most of them actually, as you can see, it's in the UVC range. Now, we thought we didn't have UVC light on the planet, right? That's not, according to NASA, that's not what penetrates. But it turns out that we make that in our bodies. We make all sorts of light, including UVC, and that's what actually activates that. But we need to receive the sunlight and the infrared light and all the spectra of the light in order to be able to do that. So another jump out, this is something that Logan wanted to really need to add, but the aromatic amino acid connection is so important because tryptophan and serotonin both absorb UV light, and as you know, tryptophan produces the precursor for serotonin, and 90% of that is made in the gut. And so it's very important. We were trying to connect that with the gut versus the brain type of Parkinson's, and we think the gut is also a light issue, and it is actually the light that is not, or it is being made by the microbiome, and maybe one of the microbiome researchers can actually look into that piece of what role that light plays in the gut, in these signaling molecules, and in the neuro and gasotransmitters. So it will be very interesting to dive in that. This is a paper I found published in Chemical Science in 2020, and they actually have a new interpretation of the Raper-Mason pathway, which was designed 70 years ago, that talks about how melanin forms the different polymers, and they actually said that the interpretation should be changed and updated because it really depends upon what type of melanin you form, depends on the local environment. And that's of course also toxins, but also light, we think. So the positive role for UV light is very much underappreciated, and this beautiful paper highlights that as well, that truly ultraviolet radiation regulates the brain and the immune system. So loss and dysfunction of melanin. As we know, in the brain, we know what happens. Insomnia, sleep apnea, is all part of the Parkinson picture as well. We get cataracts, myopia. In the ears, I don't know if many people relate that, but tinnitus or tinnitus and Meniere's disease is also related to a loss of melanin in the ear. Partial or total loss of smell, that's related to it. And then we have the skin issues. Vitiligo is thought to be the process of your body trying to realize we don't have enough not enough dopamine, we don't have enough melanin, we need to pull it in and push it, get that melanin used up. And so sunlight can actually reverse that. And then the spinal cord and gut constipation and adhesions are also thought to play a role. Now, this is another piece that I think is crucial, which is time. And the light you receive has to be very much precise on the circadian rhythms. That sets all the tone. That gets all those, these are all genes that are clock genes that clearly signal what has to happen in the body and how it all takes place. And so this is a beautiful paper called Fighting Against the Clock, the Circadian Disruption and Parkinson's Disease. And so another piece of the light puzzle. Here they did a beautiful job with this diagram clearly showing how it affects the gut-brain axis, neuroinflammation, mitochondrial dysfunction, and oxidative stress. And then 5G is another huge piece. I brought my meter with me, so if anybody would like to see how their devices might be radiating on them, I'm happy to show you. So it's another huge piece. What we learned is that it dehydrates the melanin, and it also inhibits production of the enzymes that are required to produce melanin from POMC. So that's a whole other story. There's an amazing paper that it was over 1,000 pages in 2020 published. That's the true extent of the severity has been grossly underestimated of all of these frequencies that we are bathed in every single day. And then a brief slide about the energetics of food. Matthew Phillips is the metabolic neurologist, and he did a study in 2018 comparing the low-fat versus high-fat diet for Parkinson's patients, and he found that there's definitely a benefit for consuming a high-fat diet. And ultimately that is because food is simply broken down in electrons and protons, and the deuterium content of that food is determines your quality of the water, the metabolic water you produce, and the quantity of it. And so we know that with every molecule of metabolic water we make, we also produce 2 electrons that can then help maintain the net negative charge of the cell. Points for future integration. These are parts that we also wanted to look at but had no time to explore any further. But we hypothesized that because of blue light that actually separates retinol from melanopsin, and that then destroys many different opsins in the body, and that then another cascade of reactions that takes place, destroying melanin sheets, destroying all the heme proteins. Of course, we need the P450 proteins for detox. We need hemoglobin. We need it for all the chromophores. We need it for everything. And so that gets diminished. What happens then is the mitochondria fail to emit the coherent light. This is Van Wyck. I really encourage you to look at his book called Light Shaping Life, which is right here. We can have a little show and tell. Take a look at it. It is completely filled with Martha's millions of notes. And then the integrity of the endothelial glycocalyx fails. And then heteroplasmy, that's a term by Douglas Wallace, a mitochondrial researcher at CHOP, and entropy that increases. Cells become more vulnerable to excess deuterium, and the fungi exploit the chaos as a downstream effect, and on it goes. So finally, then what do? And we want to restore through light and melanin renovation. And so these are all the pieces that we think are key. So exposure to early morning sunlight, and if you can, please also see the sunset. UVA and infrared light spectra throughout the day. So sunlight is always 42% infrared light. Crucial to catch at all times of the day, but throughout the day, the sun gives us many different spectra that are also very healthy for us to be exposed to. You must try and do your best to avoid artificial light. Change the light bulbs in your home, get rid of all the compact fluorescents, buy the old incandescent candles, fireplace, those types of light. Manage your tech exposure. Work on as much as you can on your laptop. You have to be on a wired laptop on the Ethernet cable. Shut off your Wi-Fi. It's a tremendous difference, and I can show you. Drink deuterium-depleted water if you can, or at least low deuterium water and spring water. Consume largely a higher fat diet and low carbs, and optimize copper status. Cold thermogenesis, something that Jen did this morning early on. She went in, I just missed her. But that's another huge piece of restoring mitochondrial function. And then proper minerals. And so, yeah, that's it. Thank you so much. She did. I just missed her. She did it.